This is our lab for microscopic and simple staining lab report for Group 2, Rita Cantu Mahurin, myself, and Diego Castillo. In our introduction, we highlighted the importance of the microscope. It was an essential tool that allowed us to observe the microbial morphology as well as the structure. We utilize simple staining, which is a single dye, and in the case of this lab, we utilize methylene blue to enhance the contrast as well as make the bacterial cells more visible. This lab also introduced us to the use of the compound microscope, proper smear preparation, heat fixing, and simple staining. The cultures we observed were SRS, B. cereus, and S. typherium. For our procedure, although this is a condensed version of the procedure we utilized throughout the lab, this is a summarized version. So on the smear preparation, we began with a small drop of DI water after we cleaned the glass slides, and we placed this inside the wax circle, which we used the wax pencil to create the circle as well as label our glass slides. We sterilized the loop, collected a small amount of colony, and we mixed it with water. We spread the smear to a nickel size and we allowed it to air dry. In the case of our lab specifically, we actually utilized the hot plate to air dry, and then we used the Bunsen burner to heat fix the slide in comparison to the incinerator platform that was provided in the procedure because we do not have that in our lab. The simple staining included placing the smear on the staining tray. We would flood the glass slide with methylene blue for a minute. We would rinse it gently with water and would blot dry it. And then we would observe it under the compound microscope. And if we were to use the 100 times objective, we had to use the oil. And it was imperative that we clean the microscope after each use to avoid contamination, specifically between the 40 times objective and the oil.
These are the results for SRS. It shows a bluish color from the methylene blue, and it is a spherical type shape, um, which is also known as a cockeye. Um, it's good to use a thin layer of the bacteria when staining because you don't want to create large clumps of the bacteria where it's difficult to see the actual shape of it. So this way, with that thin, evenly spread layer, you'll be able to see all the different shapes of the bacteria so that way you understand what you're looking at. And as for the SRS stain that we got, you can see a cockeye shape for the bacteria, which is great because it shows us what shape we're looking at. These are the results for B. cereus. You can see again a bluish tint for the bacteria and also some gray um, debris that could be either a result of overheating. But overall, you can still see the rod-shaped bacteria throughout the stain. Staining is very important in seeing bacteria clearer. It'll provide color. Further along the line, there will be more staining methods that use other dyes that'll have specific purposes. Um, and for this one, you can just see the color and the shape of the bacteria. So the methylene blue gave it a blue color, and you can see a rod-shaped bacteria for B. cereus. For S. typhimurium, this was our worst one, and that was on me. I feel like either I heated it too long or too little. If I heated it too little, that means that the bacteria didn't stay on the slide and eventually was washed off. Or if I heated it too much, then the bacteria just burned up and was lost. And that's why I included a picture of what it should actually look like, which is a it's a rod-shaped bacteria, and you can see the color is pink. They probably used a different dye. Since we used methylene blue, it would have been a bluish color. And the importance of the simple staining is that you should be able to see the shape and what the bacteria should look like. It won't give you any other context for if it's gram negative, gram positive, but what its main purpose is that it's going to show you the shape of the bacteria, and that's what you really want. As you can see from my results, there's nothing there. So that's the importance of our staining and our heat fixing, because if not, then you're not going to have anything and you're not going to see anything. But when you dye it, you're going to be able to see, like in the picture at the bottom, you're going to be able to see the bacteria specifically, and it'll reveal shapes that you need in order to identify what you're looking at, because you can get different types of shapes for bacteria, whether it's rod-shaped cocci, staphylococcus, where it's um, altogether. There's just different things that you should factor in.
So for the conclusion, the purpose of this lab was overall to gain familiarity with the compound microscope and practice simple staining so that we could visualize the bacterial morphology. By utilizing the methylene blue, we were able to increase the contrast between the bacterial cells in the background, which helped us more clearly identify the cell shape, size, and arrangement. This lab also gave us practice with the critical preparation steps, such as making a smear, air drying, and heat fixing before staining. Each of these steps is essential for obtaining accurate results. In the particular portion of heat fixation, it's crucial because it kills the bacteria, so it preserves their structure and ensures that the cells adhere to the slide throughout the staining process. One area of difficulty we encountered was the heat fixation step. We struggled to apply enough heat for a sufficient duration, which caused some of the bacteria to wash off the slide during staining. So as a result, it was challenging to view the cells clearly under the microscope. This demonstrated how small errors in technique, like inadequate heat fixation, can reduce the ability to properly analyze a sample. Overall, this lab reinforced the importance of careful technique in microbiology. Even foundational skills like smear preparation and heat fixation can directly affect the success of staining and the quality of microscopic observations. Thank you. We appreciate your time. This was once again Group 2, Diego Castillo and Rita Cantu Mahurin. Thank you.